What the alternative pathway does and why you should care, one of 3 complement activation pathways. First, let's introduce some key players. C3, the complement component 3, C3A, a chemoattractant for phagocytes, C3B, binds to pathogen surfaces and tags it for phagocytosis, IC3, a hydrolyzed form of C3. Factor B, a plasma protein, BA and BB, components of factor B. IC3BB cleaves C3 into C3A and C3B. This is known as a C3 convertase. C3BB, a membrane-bound version of IC3BB. Factor P increases C3 activation. Factor H decreases C3 activation. Factor I works with factor H to decrease C3 activation. Initiation of the alternative pathway is triggered when C3 is near a pathogen. The unfamiliar environment of a pathogen surface accelerates the spontaneous C3 conformation change that exposes a highly reactive thioester bond. That thioester bond undergoes a reaction with water, forming IC3. IC3 binds to factor B. Factor D comes along and cleaves the factor B portion in two, producing BA and BB. BB remains attached to the IC3 while BA remains free. The IC3BB complex is then able to cleave C3 into C3A and C3B. Exposure of the thioester bond in C3 activates C3. Activation of C3 does not only occur near a pathogen surface. The thioester bond is exposed spontaneously as the C3 molecule travels throughout the body and is exposed in response to the high concentration of C3 in the blood. How does our body keep this pathway in check? Complement activation is increased by factor P. Factor P binds the C3BB complex and protects it from protease degradation. Complement activation is decreased in several ways. Factor H binds to C3BB and works with factor I to cleave C3BB from the pathogen surface, preventing further cleavage of C3 by that complex. Decay accelerating factor, also known as DAF, binds to the C3B portion of C3 convertase and causes the C3B to dissociate from that complex. Membrane cofactor protein, also known as MCP, works similarly to DAF while still leaving the C3B susceptible to cleavage by factor I. So, what happens after C3 is activated? C3A fragments are released into the tissues after cleavage, where they begin their work as anaphylatoxins. C3A fragments recruit phagocytes and fluids, causing inflammation. The inflammation caused by C3A can lead to the reaction known as anaphylactic shock, hence the name anaphylatoxin. Macrophages arrive at the infected tissue where the CR1 receptors on their surface bind to the C3b fragments covering the bacteria cells. This leads to the macrophages engulfing and destroying the bacteria cells. CR3 and CR4 receptors on macrophages bind to the free-floating IC3b fragments. This interaction produces the same result as the CR1 receptor and the C3b reaction. Innate immune response and the alternative pathway. The innate immune response is characterized by an immediate, robust, and non-specific reaction to invading pathogens. As demonstrated by the spontaneous activation of C3, even without a pathogen present, the alternative pathway is always ready to fight against invaders. C3 is also activated when in the presence of unfamiliar bacterial and fungal surfaces, regardless of the type of bacteria or fungus. The alternative pathway is also the first of 3 complement pathways to be activated, making it a frontline defense for our innate immune system.